Tumour suppressor gene we are going to do today. Under this competency, which describes oncogenesis. In an earlier video, we had already done oncogenes and their activation and also focus on P53, made a full video on that. So tumor suppressor gene. So all of you know that cancer is a disease of genome. And there are two types of molecular accidents which can alter the genome in such a way so as to lead to cancerous transformation. To drive the cell to malignancy, two types of molecular accidents are there. Either activation of growth promoting genes. The proto-oncogene to oncogene activation, a full video was earlier made where I try to explain this complex topic. Second, either activation of growth promoting gene or inhibition of growth inhibiting genes. The growth promoting genes, as we had seen earlier, they code for pro-mitotic proteins. Okay, and what about these growth inhibiting genes? They encode for growth inhibiting product. So inhibition of tumor suppressor genes is analogous, somewhat analogous to brakes and proto-oncogenes to accelerator. A car accident, uncontrolled speed can occur either when accelerator is excessively pressed or there is failure of brakes. So similarly, molecular accidents will take place when there is failure of brake, that is inhibition of the growth inhibiting genes, which are called tumor suppressor gene. And this is also commonly referred to as loss of function mutation. And we are going to discuss it has to be homologous inactivation. So two genes we are going to study: retinoblastoma gene, abbreviated as RB, and the second, of course, P53, all of you are so very well aware of. Now, RB or the retinoblastoma, it encodes for a protein called PRB, protein RB, and it guards the G1 checkpoint. Just to refresh, G1 checkpoint का क्या मतलब है? Cell cycle proceeds in various phases. G1, this is a silent phase. S, when the synthesis or the replication occurs. And G1 checkpoint is when transition from G1 to S occurs. Entry into the synthetic phase, that is called G1 checkpoint. And as we had seen, G1 to S transition. So we are saying that RB is, it guards this point. How does it do? Let us see how does it do. How does it inhibit transition from the G1 to S phase? We'll have to go to some basics. There is a transcription factor called E2F. E2F encodes a variety of proteins, a number of proteins more than 500. So these proteins are responsible for crossing of the G1 checkpoint. You can see, you're familiar with some of the names: thymidylate synthase, DNA polymerase, topoisomerase, dihydrofolate reductase, and most importantly, I would say, cyclins, and some proto-oncogenes also. So they all are, most of them are concerned with crossing of the G1 checkpoint. So E2F क्या करता है? Let us go back. E2F is required for cell cycle progression. It enables to cross this checkpoint G1 to S. Now, E2F is prevented from activating these number of proteins by RB protein, by retinoblastoma protein. So what does PRB do? Retinoblastoma protein do? It binds with the E2F. E2F, the activator. Look at my hand movements. PRB binds with it and the binding inactivates it. Just आपको याद करा दें, all the transcription factors they have got a DNA binding domain. PRB क्या करता है, इसके DNA binding domain को mask conceal कर देता है. So E2F is not able to activate all this, resulting in inhibition of G1 to S transition को inhibit हो गया. This is how PRB acts. अब अगर मान लो, जस्ट एज्यूम, जस्ट विजुअलाइज, अगर हम इन दोनों को डिसोसिएट कर दें, अगर हम पीआरबी को रिमूव कर दें, रिमूव, so now E2F becomes activated, it gets free, checkpoint को क्रॉस किया जा सकेगा. And that is what precisely cyclin D does. Cyclins, as you people know, are certain proteins which come and go during progression of the cell cycle. तो ये सेल साइकिल प्रोग्रेशन के लिए बहुत इंपॉर्टेंट प्रोटीन्स हैं एंड दे एक्ट अलोंग विद साइक्लिन डिपेंडेंट काइनेजेस. तो अगर साइक्लिन इसको डिसोसिएट कर दे तो सेल साइकिल हो जाएगा. सो व्हाट डज इट मीन? Cyclin D is the ultimate puppet master which enables G1 to S crossing. But how does it act? Let us see. Cyclin D, puppet master, pushes the cell across G1 checkpoint. But how does it act? Normally, you know, cyclin D, it is in the hypophosphorylated form. I'm showing this circle is the phosphate group. तो बहुत कम होता है. So cyclin D, this is actually, sorry, RB protein. RB, our anti-oncogene. What cyclin D does? It converts hypophosphorylated to hyperphosphorylated. It causes extensive phosphorylation of the retinoblastoma protein. Now an important statement, the phosphorylated PRB is unable to bind the E2F. तो कैसे इसने किया? साइक्लिन डी ने हाइपरफॉस्फोरिलेशन कर दिया PRB का, so that it gets dissociated. Rest we have already done. Now one point, important point, puppet master we said is cyclin D, but ultimate puppet master I would say are mitogens. Why? Because cyclin D are under control of mitogens, a number of them you know. So mitogens, they appear late in the G1 phase और cyclin D को ये stimulate करते हैं. In fact, you know, cyclin D is the only cyclin which is under control of a mitogen. बाकी अपने आप spontaneous होता है. Anyway, besides the point, तो हमको ये सारा अब हो गया. So now overview, mitogenic stimulation induces synthesis of cyclin D. The first part, mitogen, cyclin D का synthesis increase हो गया. Cyclin D starts phosphorylating retinoblastoma protein. So the phosphorylated PRB phosphorylation releases transcription factor from inhibitory control. तो ये हमने देखा, phosphorylation ने inhibitory control से रिलीज कर दिया. तो E2F the potent transcription factor is now free to act, loosely saying अब वतन आजाद है, and that enabling the transcription of genes for cell cycle progression. So what would you say? Can you explain the statement? Puppet master is cyclin D along with some kinases. But ultimate puppet master, I would say, or I hope you would agree, is the transcription factor. All right. So now we come to the second anti-oncogene. The first one we have discussed, retinoblastoma protein. Second one, this is P53. तो आपको याद होगा P53, that was guardian of the G1 checkpoint. This is guardian of the genome. How does it act? तो अगर आप ये देख लीजिए, आई वॉन्ट गो इनटू डिटेल्स, साइक्लिन डी इज द पपेट मास्टर, और अगर ये साइक्लिन डी ही इनहिबिट हो जाए, then what will happen? This progression will be blocked. All right. This is what P53 does. This is what P53 does. The cell is to make all and none decision. हमने इसे सारा किया. So चलो anyway, I won't repeat it. Cyclin D का inhibitor. Rest of the events are same. So P53 हमने जैसे देखा था, it is the ultimate integrator of DNA damage response. It comes into picture only when after the DNA is damaged. How does that happen? ये थोड़ा सा हम देख लेते हैं. I had omitted this. जैसे ही DNA damage होगा, some sensor kinases, they get activated. So sensor kinases further transmit the signal to checkpoint kinases. Checkpoint kinases 2. And these checkpoint kinases called stabilization of P53. So P53, a nuclear phosphoprotein, जिसकी half life T करीब 1 hour है. These are the latest figures. तो इसको ये स्टेबलाइज कर देता है, इसकी हाफ लाइफ इंक्रीज हो जाती है, इंट्रा न्यूक्लियर कॉन्सेंट्रेशन इंक्रीज हो जाती है. तो डीएनए डैमेज P53 gets activated, intranuclear concentration increases and inhibits cyclin D. Rest of the events are same, leading to block here. But this question is for the inquisitive minds, for the postgraduates, how damage is causing increased half-life of P53. P53 की half-life बहुत कम है. Reason, it binds with a proto-oncogene called MDM2. Fast binding, and MDM2 very quickly sends the P53 to the proteosomes for degradation. अब ये काइनेजेस क्या करते हैं? दे कॉज फॉस्फोरिलेशन ऑफ P53. Up to 12 sites are phosphorylated. So now an important statement. Phosphorylated P53 is not a easy substrate for MDM2. So ये P53, MDM2, my hand moments. So these are sent to degradation to proteasomes very fast. But phosphorylation हो गया, phosphorylation keeps away MDM2. तो इससे क्या होगा? P53 की half life increase हो जाएगी. So that's it, I think. So key integrator of DNA damage response, guardian of genome, because threefold action delays replication by causing cell cycle arrest through acting through retinoblastoma protein, and then of course we had discussed repair apoptosis भी ये करता है. So these are the two most important ones Now we are going to summarize, and there are some more also which are not necessary for undergraduates, but the inquisitive minds and postgraduates can see these also. Some more examples of tumor suppressor gene. One, PRB, retinoblastoma protein, हमने देख चुके हैं. It is a negative regulator of GNA, G1 checkpoint. दूसरा, proteins that respond to DNA damage, of course, P53. तो इसकी inherited होने से होता है रेटिनोब्लास्टोमा, इट्स एन आई ट्यूमर. और दूसरा, इसकी P53 का हमने लास्ट टाइम भी देखा था, मोस्ट कॉमनली म्यूटेटेड TSC इन स्पॉन्टेनियस कैंसर. अ वास्ट में variety of cancers occur because of mutation in P53. एक और है नेगेटिव मॉड्यूलेटर ऑफ माइटोजेनिक सिग्नल. हमने अभी देखा था अल्टीमेट पपेट मास्टर कौन था, माइटोजेनिक सिग्नल, जिसने साइक्लिन को स्टिमुलेट किया था बाय अ सीरीज ऑफ इवेंट्स कॉल्ड माइटोजेनिक कैस्केड. फॉरगेट अबाउट इट. तो अगर ये माइटोजेनिक सिग्नल ही डिफेक्टिव हो गया तो उससे भी ये हो सकता है. तो ये NF1, this is causing disruption of the mitogenic signal, and the disease called is neurofibromatosis, inherited disease, or it encodes for a protein called neurofibromin. Much the same way, P53 codes for P protein P53, ये कोड करता है PRB. तो ये आप देख लीजिए, न्यूरोफाइब्रोमिन इज द प्रोटीन, और इसके जीन का कुछ नाम है which I am not saying for the sake of simplicity. Actually, I have forgotten also. Then there are certain set adhesion molecule. All of you know that contact inhibition is an important deterrent to malignant transformation. और अगर ये contact inhibition lose हो जाए तो then क्या होगा, malignancy. The cell will be driven to malignancy. So this is the gene located on 16q. Please find out where these two genes are located. So this is, I think, Q. Short arm of, I don't know, but I think it is short arm of chromosome 16 and gastric cancer. So these, I think, two more examples are given, I have given, and few more are also there. You can see yourself. Now, one thing, inherited disease. One point, last one, but very important, retinoblastoma. जैसे for example, there are two genes. All of you know, wherever it is located. Both these genes are encoding a tumor normal gene product. I'm going to write NGP, normal gene product. This is also encoding for a normal gene product. Of course, it is PRB. This is a tumor suppressor protein. अगर एक म्यूटेशन हो जाए, single hit mutation, the cell will not undergo oncogenic transformation. Interpretation. A single gene is, it still encodes sufficient NGP, sufficient tumor suppressor gene to to prevent cancer transformation. So doesn't it mean that they are very active, very strong, very potent? Proto-oncogene में ऐसा नहीं होता. Single mutation is sufficient. So anti-oncogenes are more potent compared to proto-oncogene. I end on a frivolous note. Negative feelings, they are always, negativity, often it eclipses positive feelings. So anti-oncogene, more potent, but that is for benefit of the body. Thank you so much. Thanks a lot. तो मिस्टर संदीप इज हेल्पिंग मी, प्लीज खत्म कर दो इसको। देख लो कितने मिनट का है।